Hello, my name is Alexandra Milan, and today I will be discussing with you Chapter 4.1, Origin of Living Cells on Earth. But first, let's discuss some key concepts. Cell biology. Cell biology is the study of individual cells and their interactions. The objective of cell biology is actually to understand the basic features of cells and know how to apply them in the treatment of diseases and such. Cell theory are basically the rules that are in place for cells to be cells. All living organisms are composed of one or more cells. Cells are the smallest unit of life, and new cells only come from pre-existing cells by cell division. Those are pretty self-explanatory. Then we have DNA, which we've all heard of before. It stores genetic information and is made up of nucleotides. RNA is going to be the star of the show today, is an intermediary in the process of protein synthesis, among other things, though it is important to mention it was not always those functions it had. It used to have more functions. We'll get into that soon. And proteins form the foundation for structure of living cells, and it's made up of 20 amino acids. Okay? So, the 4 stages of cell origin. Stage 1, nucleotides and amino acids were prior to the existence of cells. Stage 2, nucleotides became polymerized to form RNA and DNA and amino acids became polymerized to form proteins. Polymers became enclosed in membranes. Polymers enclosed in membranes acquire proteins that are associated with living cells. You could have read that on your own, but basically, right now it looks kind of intimidating, kind of confusing, but we're gonna break that down quickly. Let's start. It is important to remember that these 3 theories are still being widely debated. But the first, nonetheless, is the prebiotic soup. Prebiotic soup, as the name kind of hints to us, pre means before, bio means life. So it's before life, before cells themselves. Now, the term prebiotic soup is used to describe the slow accumulation of molecules in the ocean over a long period of time. Due to low oxygen levels, they did not metabolize. The second one is the extraterrestrial hypothesis. Well, this one, again, as the name tells us, it bases itself that these molecules came from outer space. They fell into Earth. But it's very heavily argued against because the heat at which those materials would have entered Earth could have destroyed the majority of those organic molecules. So it's kind of not very accepted. The next one is deep sea vents hypothesis. According to the theory, deep sea vents contain key organic molecules. These organic molecules have formed when the superheated rich waters, with metal ions and hydrogen sulfide within the cracks of Earth's crust mixed with cold waters. It's said that the molecules formed somewhere between the extreme heat of the vents and the sudden cold waters. The deep sea vents today are a place of biodiversity, provided sufficient nutrients for animals to survive without sunlight. Given that information, I guess if now they can still sustain life, why couldn't have they sustained life back then? So if it were up to me, it's either the prebiotic soup or this one. Okay, on to stage 2. In this stage, we see the formation of organic polymers. Here we see the simple organic molecules polymerized to form complex organic polymers such as DNA, RNA, or proteins. In this process, it is hypothesized to have occurred in either a solid surface, evaporating tidal pools, or more precisely in clay as John Bernal proposed. John Bernal wrote about the clay's optimal conditions to concentrate and polymerize organic molecules to inorganic crystals, which he referred to as minerals. He said they were minerals. It was tested. John Bernal's theory was tested and found to be quite probable, though some scientists still argue that they could have formed in aqueous conditions, specifically in the deep sea vents, indicating that the molecules could have stayed in the deep sea vents hypothesis. Stage 3. In stage 3, there is a new process referred as to probiotic. It is the aggregate of prebiotically produced molecules and micromolecules that acquire a boundary around them that allows them to maintain an internal chemical environment that is different from its surroundings. It gives them like a protection from what is around them. They have 4 key features that make scientists think they're the perfect preprocessors of cells. Those 4 features are a boundary, which we just discussed. The polymers inside the boundary of the probiotic, contain information. They have catalytic functions, and they have the capability of self-replication. And just think back a little bit of what we know about the cell. It's starting to sound a lot like the cell we know. Obviously, a much more simplified version. The first of the 2 are the coacervates, which are droplets that form from the association of charged polymers such as proteins, carbohydrates, or nucleic acids surrounded by water. They are surrounded by a tight skin of water molecules and possess osmotic properties. And they also perform primitive metabolic functions. But let's remember back a little so we can understand. As we know, osmosis is the spontaneous passage or diffusion of water or other solvents through a semi-permeable membrane. And if we think about it, it's key for those primitive metabolic functions because the osmotic properties give the cell a give and take of what they need. They can let go of what they don't need and bring in what they do need. And then liposomes. Liposomes are formed spontaneously when certain types of lipids dissolve in water. Liposomes are vesicles surrounded by a phospholipid bilayer. Within the layer of they can enclose RNA, which could explain the emergence of cell-like structures. Next, we have stage 4. Stage 4 is when we finally discuss that RNA could have been the first micromolecule. It is key and it has many functions that at the beginning could have performed all the other functions of DNA and protein. Let's get into that right now. RNA has the ability to store information just as DNA does. Its nucleotide sequence has the capability of self-replication, and RNA can perform a variety of catalytic functions, as we know are produced right now by proteins. What is chemical selection, which we need to know why? Without chemical selection, RNA would still be RNA. So let's see what happened. Chemical selection occurs when chemicals within a mixture has a special property that causes it to increase in number compared to other chemical mixtures. Now think back a little bit. Remember natural selection? When we have a certain gene or certain trait we want to carry out, we want to overpower another one, That's happening now with chemical selection. Chemical selection is picking the best chemicals and increasing them over the not so great. Then comes chemical evolution, which is the population of molecules that changes over time and becomes a new population with different chemical compositions. That is what changes the actual chemical and makes it kind of how do I say, picks the best things and then carries on with only the best traits. As happened with evolution itself. Let's go to the next one. RNA world is a hypothetical period on early Earth when both the information needed for life and catalytic activity of cells were contained solely in RNA molecules. We call it RNA world because only RNA existed in that world. RNA had to perform basically all the functions in the cell, the pre-cell. And then eventually, due to chemical evolution, RNA divided its roles into proteins and DNAs as we know it now. By creating DNA, RNA would relieve itself of storing information. Then DNA only focused on that information and strengthened its strands by making them harder to break. RNA strands used to be weaker. And then RNA gave proteins its catalytical properties. And protein, as we know, has 20 amino acids, which gives them the better catalytic ability, even though RNA still performs some cells' catalytic functions. So knowing all of this, we can kind of see that without RNA, the cells we know it would be much, much different or none at all. RNA basically was the pioneer of all its functions and then later divided it down. And that is all. Thank you for your time. I hope that helped. I hope that